Uh, Good evening, everyone. It's a regular event the college was organizing as called Evening of Difference, where we talk about different things away from medicine. We were doing some beyond medicine lectures as well as book clubs during the last few years. And if you are interested, we have covered a lot of interesting topics, and they are on our YouTube channel. So it's something I think you should be going and viewing them. Very much away from medicine. Some are actually done by the doctors themselves, and then some of them are experts in other fields. So today's event is a prelude to the tomorrow's combined regional meeting, which we are having at Anuradhapura Clinical Society. And I'm very much thankful for the members of CCP who are here in Anuradhapura helping us to organize this event as well. And so I hope that you all enjoy the evening and learn a little bit beyond medicine. And to introduce today's speaker, I would like to invite Professor Sisira Sribadana, Professor of Medicine, University of Rajarata, Faculty of Medicine, University of Rajarata, to take the podium. Over to you, Sisira.
Thank you, Dr. Dumindan Munidasa, President, College of Physicians, all the physicians who have come from far away, welcome to Anuradhapura, including some of my old friends and not so old friends. And all the physicians and their families from Anuradhapura, we welcome you. It's a pleasure to introduce Anjana. I know him for last 14 years. Although I thought I know him, I'm not sure I know him very much. Anjana is a proud product of Peradeniya. He had his PhD done at Monash University on snakebite. He is probably the most renowned expert on snakebite envenomation medical aspects in Sri Lanka. I don't want to talk too much about it. There are a lot of things to read here. However, I really don't know about his wild side. I'm not sure that I know about it. I should ask about his wife, from his wife and his ex-girlfriends, about his wildlife or wild side. Introducing Anjana, please come, researching on neglected wonders of nature. Thank you.
Very good evening to all of you, and thank you very much, Professor Siribadana, for the entertaining introduction. And I really thank the Ceylon College of Physicians, especially the president of Ceylon College of Physicians, Dr. Duminda Munidasa, and the Council of CCP for giving me this great opportunity to give a talk that is very close to my heart. I have been kind of bored with various talks related to snake bites and venom and envenoming. But this is talking about nature is something missing in my life for a significant time. So I take this opportunity to discuss with you a few things that I learned on nature. So I will be planning this talk in two chapters. The first chapter will be basically talking about the neglected biodiversity of Sri Lanka, and the second chapter about my contributions and what I have done in this field. So now if I start talking about nature, observing nature photography, going to the wilderness, going to wildlife parks and so on, things that come to many minds would be, maybe a leopard gracefully crossing a gravel road of Yala or Vilpattu, and probably herds of elephants bathing in lakes, deer, colorful birds and so on. So there's so much emphasis on these large mammals and birds, because people would like to portray wildlife as with these animals. But due to that reason, if you go to any wildlife park in a holiday, you see jam-packed wildlife parks, too many vehicles, people are so much interested on these animals. But is that all what we have as a country when it comes to wilderness? So what about, the dense rainforest we have in the wet zone? And no one would probably bother or interested on maybe a tiny little frog calling at night in a dense rainforest. Maybe colorful fish that swim in streams of these rainforests. We have not only that, we have cloud forests in our central highland. So there are so many places. So my talk is mainly regarding frogs, lizards, snakes, fish, and other small animals. So just give a bit of a brief introduction. Now, what is biodiversity? Biodiversity is just the variety of life on Earth, plants and animals. So this diversity occurs in 3 levels. One is genetic diversity, especially within species. And then species diversity, that is how different the species are. And then ecosystem diversity. What about the habitats? What about the diversity of the habitats? So a habitat in a coastal region is different to a habitat with mangroves. And it is different to something in rainforests and something in the grasslands, something in cloud forests. So they all differ. Now, globally, regions with richest biologically or biodiversity high regions and most threatened regions are recognized as biodiversity hotspots. So there are 34 biodiversity hotspots recognized in the world. Basically, the qualifying characters are you should have more than 1,500 species of vascular plants, and also about 70% of the natural vegetation, that is the primary vegetation, should have been lost. So that's at a danger. So of these 34 biodiversity hotspots in the world, Sri Lanka and Western Ghats of India are considered one biodiversity hotspot. Sri Lanka alone would not make a biodiversity hotspot, but together with Western Ghats of India, we are called a biodiversity hotspot. Now, if we go back to the beginnings of the Earth, which occurred about, we believe it's 4.5 billion years ago. That is 4,500 million years ago. Early days, Earth was not suitable to life. It took another 3 billion years to develop, or the first animals to appear. So animals are quite recent on this Earth. And then first vertebrates appeared about 380 million years ago, and first mammals about 250 million. And humans, us as a species, is very, very recent, only 2.5 million years. Actually, when I call humans, it's not a single species, genus Homo. So we are animals of genus Homo, species Sapiens, but there are lot many others. Now, if I summarize everything happened on this Earth to 24 hours, that is, origin of the Earth starts at 12 midnight, and the time that I'm speaking to you at 12 midnight next day, humans evolved somewhere around 11:58, 43 seconds. And actually the sexual reproduction, even in single cell organisms, that appeared only at around 6 p.m. And mammals evolved at 11:39 p.m. So you understand, considering the age of the Earth, how recent we are. And the problem is humans are a pretty dangerous species and a very, very aggressive species. So if you go back to the previous slide still, you see now during this 4.5 billion years, there are a countless number of species evolved on Earth. And many of them perished. They were not able to survive with the changing environment or changing circumstances. So they perished. So what we see as animals and what we see in the surroundings, plants and animals, they are just a snapshot of dynamic processes happening throughout the history. And certain periods of the time, there were mass numbers of animals suddenly disappeared from Earth. We call them mass extinctions. So there were 5 mass extinctions that happened before. And the reason one was led to the disappearance or the perishing of the large reptiles like dinosaurs. We believe that is because of meteorite hitting or maybe volcanic activities. But now, at the moment, we are in the 6th mass extinction, which is driven by humans. So once humans appeared on Earth, suddenly the other animals start perishing because we as a species changing everything according to what we like. We select which species to survive, which species to perish. We select certain species, okay, this is our food, we will raise them. And then we will cut down forests and we will grow the food that we eat. So that's called monoculture. We are getting rid of the diversity of plants, but we are instead, we grow what we like. And especially after this industrial revolution, this extinction rates of other species have they are being accelerated. So large number of species are now being extinct. Now, there's one thing that we usually haven't understood clearly, I guess, at least me, during my A-level biology, or I'm belong to the last batch who did 4 subjects. So then I think after that, especially the newer consultants, they probably have done 3 subjects, but I did 4 subjects. So one thing that we misunderstood was that, Australopithecus afarensis, from that Homo habilis, Homo erectus, and then it became Homo neanderthal, and then Homo sapiens, which is not the case. So different types of humans, that is, species belonging to genus human, Homo, existed on Earth different times. For example, 38,000 years ago, when Balangodu man lived in Kuruita Batanabalena, same period in Europe, Homo neanderthalensis was living in harsh, colder environments in Europe. At the same day, same era, in Indonesian tropical rainforests, there was a species called Homo florescensis, which is about 1 meter height, 25 kilogram human species. And in China, Homo erectus was still living. Homo erectus survived for 2 million years, roughly. But now, at the end of the day, it's us, Homo sapiens, surviving. Probably because we were more competitive, we had our ability to, more higher cognition ability, and we have that ability to think like a group, organize like a larger group, not as small groups like what others did. And also, we were able to creatively come up with solutions for various problems. For example, to catch a fish, a Neanderthal would, although Neanderthals are quite big, we believe they are quite physically strong, large-bodied ones who are, they can survive harsh physical environments. But as a species, Homo sapiens was able to, come up with creative ideas to catch animals, catch fish. So that's why we survived. So one other important thing that we need to realize is that even from the earlier days when humans started, how that affected other animals, the existence of other animals. Now, when Balangodu man is a anatomically modern and culturally modern human. So basically, anatomically modern in the sense it has all the modern characteristics in Balangodu man. So Balangodu man hunted various other animals and and in his caves there are bone evidence of other animals. So, for example, Sri Lanka, we had our own lion, which perished about 100,000 years ago, which is a big lion species, compared to the Indian one. And then we had our tiger, which lived up to like 14,000 years ago. And definitely the bones of our own tiger, this was there in Balangoda Man's caves. Probably, we don't know whether Balangoda Man ate the tiger or maybe whether it's a part of rituals that they performed. And suddenly we had two species of rhinoceros, single horn rhinos, one species of hippopotamus, and a gaur, a large bull, which existed until 1800s. When the British were here, the gaur was there, but gaur still survives in India. So likewise. So when you think about the story of biodiversity, especially in the recent past, it's amazing how we lost certain species. Now, getting back to Western Ghats and Sri Lanka, this biodiversity hotspot has more than 5,000 flowering plants, 139 mammals, 500 bird species, and 179 amphibians, and many of them are critically endangered. But one thing we don't appreciate is that now, although we consider Sri Lanka and India, the Western Ghats as a single biodiversity hotspots, there are a lot of differences between animals in Western Ghats of India and Sri Lanka. Now, that is because now if I consider the last 500,000 years, the time periods we and India were united as a single landmass was more than the time we were separated. So the last time when the sea level rose, I think about 20,000 years ago, we started separating. So if you look at this graph, the bottom one, this is 0 is present day and 450 is 450,000 years ago. And this 0 is the current sea level. And this line represents about 65 meters below the current sea level. So the eras or periods shaded in gray are the periods where sea level dropped below 65 meters. So you see, every time it drops the sea level, the contour of the map looks like this, or even greater than that. So there's like across the Pork Strait, there was about like 100-kilometer wide land bridge connecting India and Sri Lanka. So last time we were separated during the last ice age, that is about like 20,000 years ago. So during all this period where we were a single landmass, animals freely crossed from India and then vice versa as well. And during the times of separation, these animals would evolve into to their own niche of habitats within us, so that there are similarities as well as there's a lot of differences because of repeated separations and the unifications. So because of that, especially the small animals like frogs, reptiles, especially the snakes, and then fish, which cannot cross this, this dry land barrier or the dry strip of land. They were forced to stay in Sri Lanka or restricted into our wet zone and evolved as different species. And species that were able to cross, they freely migrated. That is how it happened. So, therefore, although we belong to the same biodiversity hotspot, Sri Lanka has enormous diversity in the wilderness. So we as a country has the highest species density when it comes to flowering plants, amphibians, reptiles, and mammals all throughout Asia. However, we only know about a very little of Sri Lanka's biodiversity, especially considering the invertebrates and other small groups of animals. And of course, about like 30 odd frog species that were extinct, known to be extinct from the world over the last 200 years, 18 were from Sri Lanka. Because we know about them because in British Museum and other European museums, early Europeans said, the planters, they have collected specimens and deposited there. Now we go there and when we examine these specimens, there are no matching living populations in the country because they were extinct and gone. So now the study, scientific field that studies these, the differences of these animals or the exploring the organisms are called systematic biology. Taxonomy is part of that. So basically what we do is, in this science, we identify organisms and appreciate their differences with other animals, and then think about examine the morphological differences and genetic differences, and also investigate their evolutionary history. So that is the important part for any form of conservation. Before conserving animals, we have to identify which animals do exist. So interesting thing is, when it comes to systematic biology of animals of Sri Lanka, those who have researched are mainly doctors, medical doctors, although they are not trained zoologists. So starting from Patrick Russell, he never came to Sri Lanka. He was based in India. He was a surgeon working in British East India Company. He started first documenting animals who are sent from Sri Lanka to India. Especially, the Russell's viper was named after him, because Dr. Patrick Russell drew specimens of Russell's vipers to describe that. And then a range of scientists like Albert Gunther, Peter Bleecker, Frank Wall, they all are medically trained doctors, working as doctors, but then turned zoologists. Especially, I think L.A. Nichols was a very recent scientist, I think based on Colombo MRI and as well as Colombo Medical Faculty, as the head of the Department of Bacteriology in early, 20th century. So he was also a snake person who discovered snakes. But the iconic figure is E.F. Kellart. Kellart was born in Ceylon in 1819 from a Dutch-German family. And then he only lived 41 years. But to this very short life, he worked some amazing things on Sri Lanka's biodiversity. So he started documenting all the species. He tried to document all the species, mammals, birds, amphibians, and also snakes, and mainly invertebrates, especially the marine invertebrates. He described 101 marine invertebrates, new species, 3 snakes, 4 amphibians, 36 mammals, and 3 birds. So he's an amazing person, but unfortunately died en route to England when he was just 41 years old. His medical training was actually from Edinburgh, but then after qualifying, he came back to Sri Lanka and started working. So in Kellart's book, this book was published in 1853. So in his book, he mentions every great thing is composed of many things that are small. In Latin, nihil est aliud magnum quam multa minuta. So basically that shows how he is interested on these minor small things. So into my chapter 2, that is my story of getting inspired by this and getting into exploring these wonders. So from the school days, I was quite interested on these small animals. I used to have a vivarium at my room. That is a small fish tank turned into a small, inside that small fish tank, there was a small water collection, as well as there's some logs as well as some soil. So that I used to bring frogs and keep in the tank so that frogs can breed. So every afternoon, I take a water spray gun and spray the frogs so that frogs think there's raining in the forest. So they climb up to the plants and start calling. So there are female frogs. They also then get close to them because of the calling. So these things actually really inspired me. And then I started documenting these animals, what I see them, and I used various identification books. But my knowledge was not very good enough to go beyond what we learned from A-levels. A-level zoology. So after finishing A-level, I met one person, Mr. Dharmasri Kandambi, who was the curator of National Maritime Museum, Galle. So if you have been to Galle Ramparts, there's a maritime museum in Galle, and he was the curator. So he is a person who understands the diversity of these animals, and he knew how to differentiate different species of frogs, different species of birds, different species of fish, and so on. And he had a small lab with a microscope and specimens, because it's museum. He used to keep museum specimens. So me and my friend Kalana Maduage, we together used to go after our A-levels to meet Mr. Khandambi and learn about identifying these animals from him. So Galle is a very interesting place, because in this close to Galle town, about like 15 kilometers away, there's a forest reserve called Kottava Forest, proposed forest reserve. It's a 38-acre small forest, but it's like a living lab. It has everything, all the rare animals in Kottava. So you take a bus, half an hour, you will be ending up in Kottava forest, and then you can explore whatever you want. So basically, we used to go to the forest at night, and whole night we are walking in the forest, right? Sometimes in the rain, and in the morning we take a bus and directly going to meet Mr. Khandambi with what we saw and then discuss. So likewise, we actually get a habitat touch. Habitat touch is very important, where you can just, once you see a habitat, you understand what kind of species there probably could live in. For example, if I want to see a hump-nosed viper, I get a pictorial memory of what kind of plants, what kind of leaf litter arrangement should be there for the animal to be in that habitat. And especially to understand the calls and memorize frog calls. From the calls we can say which frog is calling, likewise. So this habitat touch we got from Mr. Khandambi as well. But then after that, we both moved, selected to Peradeniya Medical Faculty and started studying medicine. But at the same time, we met probably the best biodiversity scientist Sri Lanka ever produced, Mr. Rohan Pethiyagoda, when we were like in second year. So Mr. Rohan Pethiyagoda completely changed our lives. He inspired us to study animals in a scientific way and do research to the international quality and publish in good international journals. So he helped us from the very beginning, like from our second year onwards, to do research on biodiversity. And he has established a biodiversity research institute called Wildlife Heritage Trust in early 1990s. And they had a very nice laboratory in Agrapatana, 50-acre forest, the abundant tea 50-acre land, he turned into a forest. And then in that land, they had a lab which has all the facilities for us to go and work, had a great museum collection where we can, you don't need to go to Singharaja to catch a frog in Singharaja, because there were specimens in museum so that you can sit down with a microscope and see. And also they had a very great collection of books, wildlife books. So me and Kalana spent most of our weekends and the holidays, especially in Agrapatana, visiting whenever possible. So to give a brief understanding, we'll play a small video clip about Agrapatana lab and what Mr. Rohan Pethiyagoda has did in Agrapatana. This is taken from the YouTube channel of Relax Enterprises. Relax Award is one of the major awards given for entrepreneurship in the world. Mr. Rohan Pethiyagoda won the Relax Award, and from that money also he started some of the other projects to conserve frogs.
We've got this unusual ecosystem in which we have incredibly high biodiversity, and I spent my childhood years in that landscape. As my father fished, I'd go out and look at things and wander off in the forest. Out of the 15,000 square kilometers of land in Sri Lanka's southwestern quarter, about a third is occupied by tea and rubber plantations. As a result of that, the whole of the historical biodiversity in those areas has been destroyed. We can't turn the clock back to the Garden of Eden. That's never going to happen. But if you're going to turn the clock forward, how do we save as much of the diversity as we can? I got the Rolex Award for the Agrapatana Montane Forest Restoration Initiative in the year 2000. The enhanced profile that Rolex gave me really helped me take my work forward. as these tea gardens are abandoned when they're not productive or not profitable, we have the opportunity of returning them to forest. If we do nothing, what happens is the landscape is very quickly taken up by alien species. We plant shade trees because they help the Sri Lankan species of trees to come up beneath them. They provide enough shade that those alien invasive plants can't grow any longer because they like direct sunlight. 23 years later, it is still going on and it's doing very well, and we've got something to show for it. For example, within 10 years of beginning the restoration work, we found that the majority of the amphibians in highland forests had come into the land and colonized it. They weren't there before, but they'd moved in. It's a very slow process. 300-400 years of work is going to have to go into this. This is not something that I can do single-handedly. So now I'm trying to build a cohort of younger people specialized in biology who can then take on this kind of work and take it to the future. But there were huge gaps in knowledge of Sri Lanka's biodiversity because there was no literature in the local language of 80% of the people in this country. So we had publishing literature in Sinhala as one of the priorities. And from the outset, when we started this project, we built visitor accommodation on the property so that university students, researchers from all over the world could come and work there. And now some of those people are 40 years old and they've become professors in university and they've got students themselves. So that makes me very proud. When you plant a tree, you're probably not going to live to see that grow into a giant oak. People 100 years from now can look back and see what we wrote today.
So then after that, when we were in third year in the medical school, there was a non-academic staff strike that dragged for about 1 month. So then at that time, we were basically spending time in Nagrapatana. And then I was helping them to maintain the museum specimen. So if you have a museum, that means you have to constantly check the alcohol levels of the specimen bottles. If they become too diluted or evaporated, then specimens won't live that long. So there's some maintenance work need to be done. So we were actually volunteering for maintenance work. So while doing such, I was cleaning the lizard jars. So they have jars with different types of lizards. So then while cleaning them, I realized that I was cleaning actually the lizard jars of kangaroo lizards. So there's one kangaroo lizard at that time Sri Lanka had. Kangaroo lizards are small lizards, about maximum about 12 centimeters from snout to end. With the tail, it will be around 20 centimeters. So these lizards they run fast. They are bipedal. They run with the hind limbs, with the forelimbs raised on the forest floor. Fast runners. So there was one species, thought to be one species all throughout Sri Lanka. So I was cleaning their jars, and then I realized that I can't really feel like categorizing them into one stack. I realized that there are some different lizards in the same bottles. So then when I took them out and measured, I realized that the fifth toe of one lizard is actually shorter than the others. So there were shorter fifth toe lizards and longer fifth toe lizards. And then when I really looked at the male, they have a dewlap or a gular sac, a sac on the neck for males that is to display to females. So there's a red patch in the ones with shorter, fifth toe, and black patch with the ones with shorter fifth toe, and a red patch in the ones with longer fifth toe. Then I really start to separate them. There were other features like differences in the male sexual organ and so on. And when I plotted them in the map, Those ones with short epiphyta were all from dry zone, and the ones with red patch were all from wet zone. Basically, so we were quite convinced that this is a new species that people haven't, realized or appreciated. So when I was in 4th year, this paper was published. This was my first paper, so a senior scientist, Mohammad Bahir, helped me to write it up in the proper way and document it. So we published this in a very good journal, Raffles Bulletin of Zoology. And for that one, I won the presidential award when I was just in like 4th year. And then this name that we given was Otocryptis nigri stigma. Otocryptis is the genus name. Nigri means black, stigma means patch. So a black patched Otocryptis. That's how we put that name. And then while doing 3rd year clinical appointments in Matale Hospital. Matale is an interesting place because you have all sorts of climate zones. You have dry zone close by, you have intermediate, and then you have cloud forest as well, because Knuckles is around. So Matale Hospital, when we were doing medicine appointments, our consultant was Dr. Nirmali Gunawardana at that time. So we were 3rd year students. So into the wards, we used to get a lot of hump nose viper bites. So me and Kalana tried to identify hump nose vipers using identification keys. At that time, people knew there were at least 3 species in Sri Lanka, but not sure which ones fit with the descriptions. So we soon realized that the current literature at that time was not capable of separating the 3 species or the identity. So we started a big project on investigating the hump nose vipers throughout the country. So for the next 6 years, even until we passed out, still we were carrying out this study, which was finally ended up in, I think, 2009, revising the entire genus. So we basically identified 3 species, Hypnale nipa, and Hypnale zara. So we gave the latter 2 names that is still being used at the moment. So we started this as a 3rd year project, so but published in 2009. So now with this study, we cleared the species boundaries that Hypnale lives in Sri Lanka as well as southern India, but Hypnale nipa is a highland species living in and around Nuwara Hatton area, and Hypnale zara, the other one, is a rainforest species. It lives in rainforests. So that is how we delineated the taxonomy. And then we found, while we were doing this study, we came across a new, potentially new looking species. One of our friends in Gaul, dearly friends, Amal, people from Gaul know about him. Amal is a great, naturalist who does a lot of snake rescues. When snakes come into houses, he used to voluntarily help to rescue snakes. So Amal informed us that there's an interesting looking hump nose viper, probably a new one. We haven't seen something like that. And then we quickly got the specimen. And apparently it looks completely different. And then we studied the bones and all that, sufficient enough for calling as a separate species. Unfortunately, the problem was only 1 specimen. So we can't show that there's a population surviving of this species. So we can't exclude the fact that this is a mutation or a mutant specimen. So then in that paper, we kept it open, saying that this is a potentially new species, Hypnale amal. So we didn't call it as a Hypnale amali. If we call it Hypnale amali, it becomes a valid species. So we call it Hypnale, a potential new species, amal. And then so anyone who can finds a similar specimen or population can call this a separate species called Hypnale amali. Unfortunately, Amal, who was one of our dearly friends, passed away because of a cobra bite while he was handling a rescued cobra and got bitten, and he tragically passed away several years before. So after completing MBBS, then we were thinking of what to do in the pre-intern period. So whether to join a department as a demonstrator, or what to do. And at that time, Mr. Pethiagoda said, why don't you come to Agra and work as a full-time field biologist? So we took up the challenge and went to Agrapatana and stayed 11 months, total pre-intern period in Agrapatana as field biologist. So Rohan Pethiyagoda is, although he's by profession, he's a biomedical engineer, but he's a zoologist, recognized zoologist, and he's an expert in ichthyology, that is the study on fish. So Mr. Rohan asked us to work on fish and help him with fish projects. So the 11 months we were basically doing field work all around the country, especially in the wet zone, in thick jungles, sampling fish and identifying them and describing them. So at the same time, we developed various other techniques that we can bring into from medicine to this field, especially staining techniques of bones. So that we used to, make fish specimens completely transparent and with bone staining, so that we were able to bring osteological aspects as well to species differentiations. And then we were helped with some molecular studies done by Dr. Madhav Gaskumbura. He was doing his postdoc in Boston at that time. So with combining the morphology, osteology, as well as molecular studies, we started, finding out various new species. So during this time, we were able to describe 9 new species of freshwater fish, and 2 additional species that were earlier thought to be not endemic were raised to the endemic status. So there were different types of barbs and catfishes we described throughout Sri Lanka. And even the extinct one as well. We discovered a new one, not a new one rather, unrecognized one from museum specimens in National Museum Colombo, a spiny eel, which is finally proved to be a new species or a separate species. And one of the important questions I always get when I talk about new species, people ask me, why don't you put your name to the animals that you discover? So as systematic biologists, we don't do that. We don't name animals that we discover with our name. So we name animals to honor other people. So one of the most joyful moments of naming a new species was naming a species after late Dr. Kamalika Abeyratne. So I was when I was in during this pre-intern period in Agrapatana, I was reading her biography, A Life in the Round. So this, I think many of you know about Dr. Kamalika Abeyratne, who was a senior pediatrician, and she, I think in mid-1990s, she met with an accident, road traffic accident, and then after that, a transfusion she got infected with HIV due to a transfusion. And then after that, so this is early days, the society and even the doctors were not much, the attitude towards HIV infected people were not that good. So she was so much stigmatized initially, but then she devoted her rest of life to raise the awareness on HIV and improve the lives of HIV infected people. So we were quite inspired with this story, and then we thought of naming a new species of fish after her. So in 2008, I think our paper came out. We described a new endemic freshwater fish from southwestern wet zone, Panthias kamalika. And in our papers, we have to mention or justify why we name a species after a certain person. So we specified exactly why we named after her and how it should be called. So even the common name for fish, Kami's barb, came on from the pet name her friends used to call her was Kami. So then we have to say that you have to call this species as Kami's barb, and then the scientific name. So we published that. Then after that, I was doing my internship in Gampola, and one day I got a nice letter from her husband, late Dr. Michael Abeyratne, was a pediatric surgeon himself. Once he got to know that we have named a species after Dr. Kamalika Abeyratne, actually, he wrote to us and thanked us, which was a very joyful moment where we understood how much impact we can do. Especially people die, but if you name a species after some person, it will be there for centuries, and people will be calling a species, remembering that person's legacy and work. So then I actually started working on Dundee, that is commonest fish group. If you go back to your childhood in memory, Dundees are small fish called minnows everywhere in paddy fields and all that. So it's the commonest group of fish. So we started working on Dundee. There were 3 species of Dundee at that time. And then addition to that, we were able to discover 2 new species, both endemic. One from actually from the Sabaragama University stream, one of the streams running across Sabaragama University. One day we were going there and then suddenly saw this one and sampled, and then it was found to be a new species. And another one from Morningside Singharaj. So at the end, 9 new species of fish during pre-intern period and 2 added to the endemic list. So that 11-month pre-intern period, Sri Lanka's endemic fish numbers increased by 38% during that 11-month field work. And then after that, actually, we took our different paths. Kalana, who is here, Kalana joined Peradeniya Medical Faculty and became a professor in biochemistry. And now he migrated to Australia, working there. And this me. And this is Professor Madhami Gaskumura, worked in Peradeniya, now he's a professor in systematic biology in, again, China. And this is Dr. Rohan Pettiyagoda. And Kalum Manamendra Rachi is a zoo archaeologist. He's one of the leading experts on zoo archaeology in the country. And this Professor Suama Boyagoda, she's again a leading expert now on small mammals. So now after we started our internship, the WHO officially actually kind of stopped functioning, but Mr. Rohan Pettiyagoda continues to inspire young biologists throughout the country. There are many new young biologists who got inspired by this work and now doing hard science, real science on these animals. So I moved into snake bites, venoms, and how snake venoms cause pathological alterations in humans and the antivenom. So finally, I would like to conclude with a statement by great Dr. David Attenborough. Cherish the natural world because you are part of it and you depend on it. Thank you very much.
Thank you, Anjana, for that wonderful lecture of your life work and eye-opening for all of us. It's open for discussion, questions. Yes, Jamila?
Excellent. We really enjoyed your talk. What about Kanneliya? Have you been to Kanneliya?
Yes. Kanneliya is actually a hidden gem. I mean, like nowadays people know about Kanneliya a lot, but Kanneliya is about 20 years back. It was kind of no one's attention was there. So there's lots of, not only Kanneliya, there's a forest complex called Kanneliya, Dediyagala and Nakyadeniya. They call it KDN complex, which is rich in all this diversity of plants, especially plants, as well as other animals. So we used to do a lot of field work in Kanneliya. So you had to go to Kanneliya, but it's about 2 hours bus ride from Galle. So we used to go there at night, spend whole walking in the forest and come back in the morning. Yes.
Thank you, Anton. A very inspiring lecture.
Thank you.
Lovely to see doctors venturing into fields that many of us don't even dream of. Just out of interest, what was your PhD on, and
Yeah.
What is the work that you are pursuing at the moment in terms of?
So my PhD was on neurotoxicity of snake envenoming. Basically, I was doing some clinical work as well as isolating, venom toxins that cause neuromuscular paralysis in humans. I did it in Monash, Australia, but clinical part was done here and experimental part was there. So I was basically looking at, because theoretically, if you see paralysis after a snake bite, there are different types of neurotoxins. So the presynaptically acting ones and postsynaptically acting ones. So the presynaptic ones are the most important ones for humans. So in my studies, usually I try to show that, especially using, in clinical studies, I use single fiber electromyography in snake envenom patients. And also at the same time, I did experimental studies to show what exactly happens in humans and how it differs from animals. For example, now you know this cobra and mongoose fight. So in the wild, irrespective of whether it happens in Sri Lanka or Africa, when a cobra and mongoose meets, they fight because mongoose is a snake eater. So it's the diet of mongoose. So I was quite curious on why mongoose usually don't die after get bitten by cobras. They usually survive. So the reason is actually the nicotinic acetylcholine receptors of mongoose have 2 amino acid differences from rats or mice, which is the diet of cobras. So we are in kind of the middle of that. So in my PhD, I was basically investigating that. I was mutating human nicotinic acetylcholine receptor and turning them to switching it to either mouse or mongoose to show that they are not clinically important for us. So that's what basically I did. So now coming back from Australia, I have set up a venom pharmacology lab in Rajarata. So we have we basically investigate how the antivenoms prevent or reverse various effects of snake venoms. And also I run with my clinical colleagues, senior colleagues, Professor Siri Badana and the medicine department people, Anuradhapura snake bite cohort from 2012. So both clinical as well as experimental studies. So now I don't do any wildlife ones. So that is, I'm retired from that part.
Yeah.
Congratulations on some fantastic work in your pre-intern and time before that. Very, very impressed by what you have done as a very, very young person. I know all about what you are doing in neurotoxicity. I'm not going to talk about that. I'm going to ask a question on the chapter 1 of your presentation. You're talking about the species that we had in our country. So one question that we always tend to talk about is we are called Sinhalayas, but what happened to the Sinhayas? So you're saying that there were some evidence of lions being here, which often gets mentioned, but what is the evidence
There
we had lions?
was one tooth, which is definitely a lion species, very close to the one in India. So India, they have lion. So definitely it's a lion. And that's the only one. That's the lion tooth is the only that was found in, because in Kuruvita, Ratnapur area, there's fossil beds. So in those fossil beds, definitely this evidence is there. Lion was there.
Any more questions? So in absence of any further questions, I would like to wind up this session, especially thanking Professor Anjana for spending your time with us on our invitation this better. And also I would like to thank all our guests today, all the participants today from Anuradhapura Clinical Society and Sri Lanka College of Physicians for being here and sharing the valuable time with us and coming to our program. So on behalf of the Sri Lanka College of Physicians, we would like to show appreciation by a token of appreciation to you, Anjana. So can I call upon Professor Udaya Ranawaka, one of the past presidents of Sri Lanka College of Physicians, to hand it over to you.